Okay, so we've been talking about some of the virulence factors that some selected pathogens are producing and how those particular virulence factors result in the symptomology associated with that particular disease. Toward the end of the period last time, I said that two pathogens that are fairly nasty are members of both the genus Staphylococcus and Streptococcus. And one of the reasons that they are both pretty nasty pathogens as a group is that members of those two genera are notorious for being able to produce a wide variety of different virulence factors. So take a look at Staphylococcus aureus for just a second, okay? Here are some factors that they produce that we talked about in the category of exoenzymes. They can produce a coagulase, they can produce a hyaluronidase, they can produce a staphylokinase. All of those help the organism to be more invasive. Additionally, they produce lipase enzymes. Those digest oils, allowing bacteria to move more easily, colonize the skin. Here is something of great significance: penicillinase. Penicillinase is also known as beta-lactamase. What does beta-lactamase refer to? What does the H tell you? An enzyme, okay. And what's beta-lactam refer to? It is that ring structure that is characteristically part of both penicillin and cephalosporin structure. And so these are enzymes that bacteria have evolved to break down those important antibiotics. Penicillinase breaks down specifically penicillin. Okay. What does MRSA stand for? Methicillin-resistant Staph aureus. Okay, methicillin is a type of beta-lactam antibiotic, and so these particular Staph aureus are producing an enzyme that inactivates methicillin, which normally is one of the preferred antibiotics to try to treat a staphylococcal infection. MRSA is serious stuff, and I'm sure those of you that are in the AT exercise science area are more than familiar with MRSA. One of the things we really have to try to make sure is that athletes don't get infected with these particular bacteria, because they can cause serious both cutaneous skin infections, and if they become systemic infections, if they travel via the blood throughout the body, it can be deadly. More people in the United States die from MRSA infections than die from HIV infections in the United States. That's significant. They are working on an an immunization against MRSA. We're not there yet, but that is an active area of investigation. So these organisms that produce these penicillin or beta-lactam antibiotic degrading enzymes are a serious threat. Additionally, Staph aureus can produce some enzymes that interact with blood components, like hemolysins, which lyse red blood cells. They can also produce leukocidins, which kill the white blood cells. So that is another strategy they can use to be more invasive, this way by kind of avoiding parts of your normal immune system. And then they can produce a number of toxins. Enterotoxin affects the GI tract. Exfoliative toxin causes desquamation of the skin. The skin starts peeling off in sheets and layers. It interferes with the desmosomes that hook cells together in epithelial tissue. And then this is one we'll come back and talk about a little bit later on. Toxic shocks toxin. That's a very systemic toxic disease that was in the news probably a couple of decades ago and was a serious public health problem. Here is, this is called scalded skin syndrome. This is produced by a staphylococcal infection, and this particular organism is producing one of those exfoliative toxins. As you might imagine, this is a pretty painful disease. And as that skin desquamates, then the skin is opened up to potential other secondary infections. So the staphylococcal infection is a primary infection, but then the destruction of the barrier of the protective skin can then make you prone to secondary infections by some bacteria that normally wouldn't be able to cross that particular defense barrier. Anybody recognize this? Have we ever had this before? This is impetigo. This is another staphylococcal infection. Here are some boils. Typically, these begin as infected hair follicles, and then as the hair follicle gets closed off by the accumulation of pus and debris, it can develop into these nasty-looking boils, and there are names for several of these different kinds of lesions. There are carbuncles, there are furuncles, but they're typically all the result of staph infection. And then the scourge of teenagers, acne. Acne is the result of the infection of hair follicles. And one of the things that tends to promote acne is an increased production of sebum, which is a waxy kind of lubricating substance that the skin produces. And if you start producing more sebum, that tends to favor the growth of microorganisms, and those hair follicles become infected. Sebum production seems to be accelerated by testosterone production. And so that's one of the reasons why young males, when they hit puberty, oftentimes have outbreaks of acne, is due to changes in their hormonal balance. So generally speaking, there are three types of staph infections. We've been talking now about the cutaneous or skin-related infections. And this particular slide breaks down a little bit more detail about some of the things that we've been talking about in terms of the particular disease, the symptoms, the agent that's a causative agent, and then what the virulence factors are that are involved in these particular diseases. Staphylococcus infections can also fall under the category of being non-invasive. And basically, these are primarily food intoxications. And so these are toxins that the staph produces to make you ill. The enterotoxin produced by Staph aureus is one of the major causes of food intoxication. Food intoxication is a fairly rapid onset condition. Typically about 4 hours after ingesting the toxin, you start showing symptoms. Fortunately, it tends to be pretty self-limiting, and the symptoms only last about 24 hours. Typically, you do not get a fever associated with a food intoxication. Now, if you have endotoxin poisoning, which is considered to be a food infection, remember that's caused by a different kind of molecule than a toxin. That's caused by an endotoxin, or the LPS layer, and that can cause inflammation and fever. So you get a little bit different symptoms depending upon the nature of the organism that's causing your quote-unquote food poisoning. The most serious type of Staphylococcus infection are those that become systemic. Systemic infections are transported through the blood, less commonly through the cerebrospinal fluid. A couple of examples of systemic infections that can be caused by Staphylococcus organisms are osteomyelitis. So what's this going to be affecting? Bones. Okay, this is a bone infection. Endocarditis. This is an infection of the endocardium. So the heart lining is attacked. This is very serious. This has a 50% mortality rate. And toxic shock syndrome is a systemic staphylococcus infection that is also highly lethal. A couple decades ago, there was a real spike in toxic shock syndrome being diagnosed. And public health experts were really kind of at a loss about what was causing this. The toxin is a substance that's referred to as a superantigen. And these superantigens cause a massive immune response. A big part of the immune response that is amplified is the production of chemical compounds called cytokines. The cytokines are chemical signaling molecules that allow immune cells to communicate with one another. And they have beneficial activities. But they can also do things like stimulate inflammation. And while inflammation is a protective response by your body, if it gets out of control, it can be deadly. So these superantigens induce what's referred to as a cytokine storm. And your body just starts dumping out all these massive amounts of cytokines. And that stimulates all kinds of nasty things, including a huge inflammatory response. And it can be lethal. Public health experts started to narrow things down, and what they finally figured out was the culprit in toxic shock syndrome were certain brands of super absorbent tampons. And they were creating an environment because of the material that was in the tampons that favored the growth of these Staphylococcus organisms that produce the toxic shock syndrome. So look at the number of cases back in the early 1980s, how high they were. This is when this really became a public health problem. And it was correlated with the production of this new type of tampon, these super absorbent tampons. When the Food and Drug Administration finally identified that relationship and required withdrawal of those particular tampons, you can see that over a period of time, the incidence of that particular disease dropped dramatically. But it took some time to figure out that particular relationship, and that the tampons were actually creating an environment that allowed that particular organism to grow and to thrive. Well, Streptococcus bacteria can be some pretty nasty pathogens as well. And again, if you look at a list of typical kinds of virulence factors that strep produces, they can have pretty long, impressive lists of substances as well. They can produce leukocytes, they can produce coagulances, they can produce, you name it, a lot of different virulence factors. Here's a nice, nasty, sore red throat. This is an inflammation of the pharynx, so this is called pharyngitis. Most people look at that, and especially the pus pockets on the tonsils there, and say, gee, that looks like it probably would be strep. It could be. Although about 95% of cases of pharyngitis are actually viral in nature, and only about 5% are actually caused by strep. But the strep pharyngitis is serious and does need to be treated. So that's one reason why it's really important to be able to diagnose bacterial rather than viral pharyngitis. We used to be limited to taking cultures. So you'd have to go into the doctor, get a throat swab. They'd probably plate it out on some blood agar. And one of the things they would be looking for would be beta hemolysis. That clearing of the agar because of the lysis of the red blood cell and the degradation of the hemoglobin. Pathogenic streptococci have been broken up into various groups that are called Lancefield strains. And I don't know where they got these designations or why they are these particular letters, but there are Lancefield strains A through H and K through V. Lancefield strain A is one of the more notorious types of streptococci. And one organism that is a Lancefield A strain is Streptococcus pyogenes. And that's one of the streptococcus that can cause streptococcal pharyngitis. One of the ways that these Lancefield strains differ from one another is that they have different sensitivity to different antibiotics. And one of the hallmarks of Lancefield strain A is that it's sensitive to an antibiotic called bacitracin. So if you take a blood agar plate and streak out a throat swab on it, and you get some nice beta hemolysis on it, and it's inhibited by a bacitracin disc that you put on there, that's pretty indicative that that is Lancefield strain A, and therefore very likely Streptococcus pyogenes. Well, now we have antibody-based rapid strep tests, and you can get the results in 30 minutes or less. And if you have truly a bacterial pharyngitis, then you can go on an antibiotic. But it's important to figure that out, because if you don't treat properly these streptococcal pharyngitis infections, then they can manifest themselves into more serious diseases. Over on the left, you can see an untreated streptococcal infection where you get a rash over the entire body. This is called scarlet fever, and this can be a consequence of an untreated strep infection. But even more serious is that untreated strep infections can also lead to a condition called rheumatic fever. Rheumatic fever causes heart damage. Why does it cause heart damage? Actually, this is an example of immune-friendly fiber. And what's happening is when you're infected with that strep bacteria, you start to produce antibodies against it. And the antibodies are specific for the strep antigens and should target them specifically. But unfortunately, some of those antigens that are on the surface of especially the mitral valve of the heart are very similar to strep antigens. And so your immune cells, in addition to attacking the strep, can attack your heart muscle as well and cause valve damage. So rheumatic fever can be a consequence of an untreated strep infection. Those consequences of other infections are called sequelae. So paralysis, for example, can be a consequence of a polio infection. Rheumatic fever can be a sequelae of a strep infection. Here is a nasty streptococcal infection that causes a disease called necrotizing fasciitis. And this is a very serious cutaneous strep infection. The skin literally just peels off in sheets. And you can see that once that skin is compromised, that this then becomes a breeding ground for other bacteria and other secondary infections. We get a lot of necrosis of the tissue, and this can be a very serious disease. So here's, again, a summary type slide that gives you some examples of some various pathogenic streptococci and various diseases that they can be involved in. But again, kind of the bottom line is that one of the reasons both strep and staph is so serious is the multiplicity of virulence factors that they produce. They produce this whole battery of substances that really aid in their pathogenesis. Okay, so what if you get an infection? What are you looking at? Well, infections typically go through kind of a standard type of pattern, which is illustrated by this particular graph of number of microorganisms and intensity of symptoms versus time. And we break up this, if you like, infection life cycle into various phases. And the first phase is the incubation phase. That's when you really have no detectable signs or symptoms. We haven't defined what those particular terms mean yet. We will do that. The infection phase, you don't really know most times that you have an infection brewing. It takes a while for the number of cells to multiply, so we can have a significant impact on the tissues. It takes a while for them to produce their virulence factors, which might be toxic to your cells of your tissues. Nevertheless, you can become infective and pass this on to other people, even though you are not showing any particular symptoms yourself. So you can be asymptomatic, and you're incubating the infectious agent, but you can still transmit it to other individuals. And that's kind of scary. Why don't we put a particular time on this particular scale? Because it's highly variable depending upon the type of infectious agent. It also depends upon the host that's infected. It depends upon their state of immunity. There's a lot of things that affect this particular time scale. Here's some incubation periods of selective infectious diseases. And I said that staphylococcal foodborne infections have a very short incubation period. Look at leprosy, 10 to 30 years. Leprosy has an incredibly long incubation phase. It is really not very communicable because it has that long incubation phase, and it's not very virulent during that incubation phase. So this disease during biblical times that was just feared really wasn't that transmissible. And when you think about the people that were put in leper colonies and things like that because of fear of other people acquiring the disease, in reality, the infection pattern didn't really bear that out. Oops. This particular period of time right here, when we start seeing an increase in both intensity of symptoms and number of cells, is called the prodromium. And this is when you have some kind of vague general symptoms. I guess it's time to define those terms. I was going to do it later. It's important to understand these two terms, signs and symptoms. signs are measurable by an observer. Symptoms are kind of general complaints that are reported by the patient. So if you tell somebody I'm warm, or even that I have a fever, that is a symptom. If your mom sticks a thermometer in your mouth and takes a reading and said you have 102 degree Fahrenheit temperature, that is measurable, quantitative. That is a sign. If you're feeling nauseous, that is a symptom. If you vomit, that's a sign. Okay, both are caused by the same underlying fact, but it differs depending upon whether it's reported by you or by the observer. The signs and symptoms associated with a particular disease is referred to as a syndrome. And so good clinical diagnosticians look at syndromes, what you're reporting, what they gather from their history, and hopefully are able to associate that with a particular pathogen that then requires a particular treatment. And that's a real art to be able to do that accurately. So in this prodromium, you have these vague general symptoms. You're feeling off. Some of that is due to the pathogen itself, but as I mentioned earlier, some of that is also due to your immune response mounting up. I mentioned that during the early stages of your immune response, you start producing some of these chemical signaling molecules, lymphocytes, including interferons. And those contribute to some of the symptomology of the illness. It's good that you produce them, but they can have some negative effects on how it makes you feel. Okay, during this illness stage now, you've got a pretty raging infection. Okay? Bacteria are reproducing rapidly. They're really in this log phase of growth. They're cranking out a lot of toxins. And so now you're starting to see some very overt both signs and symptoms associated with that particular disease. Also at this time, your immune response is kicking in. But remember that the very first time that you see a foreign pathogen, it takes time for your immune system to gear up and start producing protective antibodies. Oftentimes, that lag period means that you're going to show full symptomology and go through the full disease process. If it doesn't kill you, you will now have memory of that particular disease and a high level of immunity if you survive. So during this decline phase now, your immune system is really starting to limit the infection. If you've gone to a doctor, that could obviously be augmented by various chemotherapeutic treatments like antibiotics that the doctor's giving you. And then you get into this convalescence period, and this convalescence period, your symptomology effect pretty much disappears. But again, you can still be contagious during this convalescence period. Really anywhere throughout this life cycle of infection, you still are potentially contagious. There are some terminologies associated with infectious disease, and these are things that you really should be comfortable with. Many of these we've talked about already. The difference between an acute disease and a chronic disease is that the acute disease should be self-limiting, and you should fully recover and go into complete convalescence. Typically, in acute disease, the disease runs its course rapidly, and you have a full recovery without any sequelae. Chronic diseases typically don't have as strong of symptoms in the disease state, but they can develop slowly and last a really long time. One of the things that can be problematic is if you develop a chronic state of inflammation. Inflammation, again, is supposed to be protective. It's part of your body's protective response against infection and injury. But inflammation should be self-limiting, and it should eventually turn itself off. But sometimes it doesn't. One of the things that can cause chronic inflammation is autoimmunity. And your immune response actually attacks your own tissues. It shouldn't do that.
Where would something like you had herpes up on the screen earlier, and it said like 5 to 8 days, but can't you like get it again in like 30
Yep,
years?
herpes goes into periods of latency, and so you appear to recover, but you're always harboring that virus, and then they think that things like stress, for example, causes it to reactivate again, and again have another herpes outbreak. So unless I think they're coming up with some long-term herpes eradication treatments now, but without a treatment, you have herpes for life. So it really would be a chronic disease. It's kind of like the chickenpox virus. The chickenpox virus can lie dormant in your cells after you've had chickenpox, and then can reactivate later and manifest itself as a new disease, shingles. But they do have a shingles vaccine now. It's not 100% effective, but it's better than nothing. Rheumatoid arthritis is a particularly nasty, chronic, inflammatory, autoimmune disease. Your body is responding with its immune system against material in your joints and causing a chronic state of inflammation, which causes deterioration of those joints. A long time ago when we started this process, we talked about concepts like subacute and asymptomatic diseases. I just mentioned latent diseases, diseases that appear a long time after the infection. HIV goes through a period of latency when those lymphocytes are infected by the HIV virus. The viral genetic information is inserted into the lymphocyte's DNA, and it can sit there and last in a dormant state for a long period of time. That's why HIV has such a long incubation period, 1 to 8 years, where it's sitting there hiding in your DNA in that latent state. Not all diseases are transmissible. Some diseases are non-communicable. Tetanus is a good example of a non-communicable disease. And then there are some interesting terms associated with patterns of infection, and that's where this slide comes into play. A local infection stays primarily at the site of the original invasion. And some of those things that Staphylococcus causes that are cutaneous in nature tend to be local infections like a boil or a carbuncle, an infected hair follicle. We pretty much have the infection confined to that particular portal of entry. But if it gets into the blood or the cerebrospinal fluid, then we can get a systemic infection. I also mentioned the difference between a primary and a secondary infection. If the skin is breached by some primary infection, then it can be its effectiveness as a barrier is compromised now, and you can develop a number of secondary infections. Sometimes secondary infections are the result of treatment with antibiotics to treat a primary urinary tract infection before. So women tend to get more urinary tract infections than men do, and it's not uncommon for them to develop a secondary yeast infection after they take antibiotic for a primary urinary tract infection. So we already talked about this, the difference between signs and symptoms. If an agent is going to be infectious, it's got to not only enter the body and become established, but it also has to be able to be shed from the body so that the pathogen can then find a new host. Pathogens enter the body typically at defined portals of entry, things like hair follicles, or the lungs, or the GI tract from ingestion. These represent identifiable and common portals of exit for pathogens. And again, particular pathogens usually have a particular portal of exit. So there are a lot of different fluids and secretions of the body that can contain pathogens and can therefore spread those from one organism to another. And again, this individual may be asymptomatic and so may not know that they're doing this, but they are inadvertently shedding these pathogens even though they are, at least from all intents and purposes, asymptomatic. One of the things when we talk about infectious disease and pathogens that's important to understand is, what is the reservoir of the pathogen? Where does it live when it's not infecting a host? Where does it take up residence? Some reservoirs of pathogens are inanimate, and the most common inanimate reservoirs are soil and water. We mentioned several times that Clostridium species, including the botulinum and the tetani organisms, that the endospores of those particular bacteria are commonly found in soil. And so soil is a common source of these organisms, and that's one of the reasons why you want to make sure you wash wounds well, so you're not transferring organisms from this inanimate reservoir into living tissue. Living organisms can also be an important reservoir. Humans and animals are the primary animate reservoirs of pathogenic organisms. If you're harboring a pathogen, you could be entirely asymptomatic and still be shedding that pathogen from a portal of exit and making other people sick. You also may show overt symptoms, and you may be in the incubation stage, the convalescent stage, even the chronic carrier stage, and still be shedding those particular pathogens. Oops. There's something else associated with this. Yeah, there's part of this slide missing. I'm not sure how that happened. Chronic carriers are people who have fully recovered from the disease but show no symptoms any longer, and typically are harboring the disease in something like their gallbladder. And this is where Typhoid Mary comes into play that I mentioned earlier on. Mary had typhoid bacillus from a typhoid infection, and she recovered, but she never got rid of the typhoid bacteria in her gallbladder. And as that bile was stored in the gallbladder and secreted into the small intestine, it was contaminated with these bacillus bacteria. She was a food handler, and so if she didn't wash her hands properly, then she could contaminate food with those bacillus bacteria, and she made a lot of people sick. I think I mentioned earlier on, her story is kind of interesting. She was defiant of institutional control trying to prevent her from working in the food industry, and she worked under false names. She was institutionalized a couple of times to try to keep her out of the food industry, and she didn't really care. And she made a lot of people sick by continuing to work when she shouldn't have. Another way that humans, and this is what's missing on the slide, could cause infection is by unknowing contact in a medical setting. And so doctors and nurses, if they are carrying pathogens in an asymptomatic state, can inadvertently make other patients sick by contact, tissue to tissue. For animals, some animals serve as vectors or transfer agents of pathogen, and the animal contributes to the life cycle of the pathogen and is directly involved in the life cycle of the pathogen. When we did our survey of microorganisms, I mentioned rickettsia. And rickettsia are pathogenic bacteria that can be transmitted by arthropods. Rocky Mountain spotted fever, for example, is a rickettsial disease. So ticks and mites can be animal vectors. Oftentimes they will inject that particular pathogen through a bite, but they're harboring that particular pathogen in their tissues and are involved in its life cycle process. Other animals serve as mechanical vectors, and basically their external tissues become contaminated with pathogens. So flies are great mechanical vectors as they're flying around and getting on nasty stuff and might have pathogens. They can pick that up on their external body parts, and then they can transmit that to food or directly to the skin and cause the transmission of that particular disease. A disease is referred to as being a zoonosis if its primary reservoir is an animal, so that's its normal host. But humans can be infected by that particular agent. So humans aren't the normal host, animals are, but a human can be infected. Lyme disease is a zoonotic disease. Normal host is the white-tailed deer. But through arthropod transfer, humans can get infected with that particular spirochete bacteria. Bubonic plague wiped out most of Europe. It's a zoonotic disease. Normal host, mice and rats. But humans can be infected. The diseases don't have to be bacterial in nature. They can be viral in nature. or they can be fungal in nature. Ringworm is a zoonotic disease, normally associated with animals, but humans can catch this particular disease. These represent a lot of our new emerging infections, and pathogens seem to be gaining the ability, through mutation, of spreading their host specificity and infecting some hosts that they normally haven't been able to affect in the past. So here are some examples of some common zoonotic diseases, broken up into bacterial, viral, fungal, protozoan. Helminth diseases are worm-borne diseases. So there's a pretty impressive list of these particular diseases. What's really important with the zoonotic disease is if you want to try to eradicate that disease, you've got to identify what the reservoir of that particular disease is. And even if humans are the ones that are being infected, what you really have to do is find the animal host to treat or to eradicate if you're going to get that particular disease under control. There was a very ill-fated attempt in 2009 in Asia of trying to control the H1N1 influenza pandemic by slaughtering thousands and thousands of pigs, which they thought were the host. Pigs weren't the host. Okay? So they didn't properly identify their reservoir. If diseases are communicable, if they can be spread from one individual to another, there are typically two patterns of communication for communicable diseases. There are direct and indirect methods of transfer. Both direct and indirect methods can have both vertical and horizontal transmission. Horizontal is from an individual to an individual. Vertical is from a parent to a child, particularly mother to fetus. With one exception, direct transfer means what it means, okay? Some kind of membranes, mucous membranes typically, come into contact with one another, either by direct contact or possibly through droplets that are produced by one individual and alight on the mucous membranes of another individual. And there's a good example of a direct transmission of aerosols. This is a high-speed photograph of the sneeze, and you can see all of that mucus, which can become laden with bacteria. And if you immediately get sneezed upon, so if you were sitting over there and your face was over here, that would be considered to be that direct transmission, even though there was an actual. Okay. It's considered indirect if somebody sneezes in a room and you're not there. These particular droplets can remain suspended in the air for some period of time. And if you walk into the room later and inhale some of those droplet nuclei, that's considered to be indirect. Now, I'm not quite sure why, but they consider vector transfer to be a method of direct transfer. And that seems to be kind of a misnomer, because you've got insects transferring infectious agents from one individual to another. But according to this scheme, that's considered to be a direct method of transfer. We'll pick up here next time, because we're out of time.